Hi there. Welcome to another podcast from Week Talk Science. Thank you very, very much for joining us. Today, we have a fantastic scientist from Israel called Judith Berman. Judy, hi. Thank you very, very much for joining us.
Hi.
Could you start by telling a little bit more about yourself?
Okay, I'm a basic biologist. I started out studying yeast, mostly the model yeast, and I was interested in chromosomes and telomeres, the ends of the chromosomes, and all the different structures on the chromosomes. And I would say more than 20 years ago, by accident, actually, I got involved in studying pathogenic yeasts, and I've been doing that ever since and have never really looked back. So I did that for 25 plus years at the University of Minnesota, and over the last 7 plus years, I've been here at Tel Aviv University in Israel working on related things, mostly in pathogenic yeasts and studying their basic biology and how it influences how they cause disease and how we can treat that.
That's really interesting. So, you have actually a fantastic story to tell us today, don't you, about resistance and persistence. So, please go ahead with that concept because I think this is one thing which has got very real consequences in the real world, at least in terms of antimicrobial resistance.
Right. So we've been studying two phenomena, resistance and tolerance, in fungal pathogens, primarily in yeast fungal pathogens, which are very common and the most common causes of bloodstream infections. So bloodstream infections cause infections of internal organs, and they are associated with high levels of mortality, sometimes up to 50%. And that's in Western hospitals like in the US and Europe, where people are using the available drugs that we have. It's much worse in developing countries where, in general, they only have one of the antifungal drug classes available at all. And the difficulty with treating the fungal infections is that there are very few classes of drugs to treat them. And the reason for that is that fungi are eukaryotes, that is, they're higher organisms like us as opposed to bacteria. So their cell structure, their biochemistry, and most of their genes are very similar to ours. And so while with bacteria, we have about 6 classes of drugs that you could just treat multi-resistant Staph aureus, we only have 3 classes of drugs for treating any fungal infections, 3 classes that are used in the clinic. So if you have resistance to one of those classes that makes it impossible to use it, all of a sudden you only have 2. If you get resistance to more than one, you can end up not having any drugs available to treat the infections, and that becomes a very critical and serious problem. So my lab has been studying fungal responses to drugs for quite a while, and we've recently characterized and measured and distinguished between 2 very distinct types of responses: drug resistance, which is sort of the classical concept, and drug tolerance. And we found that they have really different properties that may provide important insights into how to treat fungal infections. So drug resistance is generally due to genetic changes, that is, mutations that make the fungus able to grow well in the drug. And these mutations generally occur in genes that either are coding for the target that the drug is attacking, or the mutations are in some proteins in the membrane that get the drug out of that cell as quickly as it came in, so that the cell is not really suffering from the drug having been around. And these are classic mechanisms of resistance that are seen also in bacterial resistance. And so once you get a mutation like that, the drug is no longer effective against the microorganism, be it a bacteria or a yeast. And basically, what this is a beautiful example of evolution happening before your eyes in real time. And you can watch it and you can study it in the lab, whereas most evolutionary processes require that you wait very, very long amounts of time to see changes. So resistance is a really great thing to be studying if you're interested in mechanisms of evolution.
Can I just ask you something real quick here?
Yeah.
So you said that you can actually watch it happen before your very eyes. What kind of frequency of mutation are we talking about, and how often and how quickly does it actually
Mhm.
occur?
So it doesn't necessarily, it's if it's it depends on the mechanism actually. So we classically think about mutations as being a change in a single nucleotide in the gene at a critical position, and those kind of mutations occur maybe once per every million or 10 million or 100 million cell divisions. But with bacteria and yeast, you have a lot of cells. So you could easily have that number of cells, within an organ or within, and certainly more than that within the GI tract, for example, of a person. So, um, so resistance is popping up all the time, or mutations that could confer resistance do pop up. Obviously, they're not super frequent, but there are other kinds of mutations which involve much larger changes in the genome, like changing the number of copies of a particular chromosome that happens to have genes on it that are important for resistance, or changes in how many copies of which copy, which allele. That is, if the fungus got one copy from its mother and one copy from its father a long time ago, and then it only has mother copies or only father copies, you can reveal mutations very quickly. And those two types of mechanism, getting extra copies or changing which copies you have, which is called loss of heterozygosity, are mechanisms that occur at about 100-fold more frequently than point mutations. And we found this one of those mechanisms, aneuploidy, a number of years ago already. Anna Selmecki, who was a grad student in my lab, did that work. And and since then, lots of other people have found that this is really a very frequent mechanism that shows up much more frequently, actually, than the point mutations, because it can happen so much more frequently and affect so many more genes at the same time when it does happen.
So when we talk about, acquisition of resistance, for example, to the drugs that we have, so here's probably a very ignorant question, but are more people dying today of fungal infections that are obviously immunocompromised than before? And I mean, how bad is that problem? What is the magnitude of this problem, basically, is I guess what I'm asking.
Well, actually, a lot of fungal infections affect immunocompromised people, either immunocompromised due to diseases like HIV/AIDS, or due to medical procedures like transplants, which require that the immune system be repressed or suppressed. And so, as medical treatments have become more used at higher frequencies, we're actually seeing more fungal infections because of that. And then, of course, because of the HIV epidemic, the level of fungal infections in sub-Saharan Africa is really high and of very great concern, especially because there are very few antifungal drugs available to do treatments.
Okay, I
So it's
understand.
becoming more frequent, not because, in part because of the medical procedures and diseases that make it more common, and also because antibiotics are used a lot, and then fungal infections pop up when the bacteria have been sort of mostly cleared and there's more room for the fungi that were already in the host to start spreading and growing without the competition from their friends, the bacteria.
Right. Okay, so and you published a paper that looked at the molecular mechanism of one part of this huge question on how fungi acquire resistance. Could you tell us a little bit more about that, please?
So the paper on acquiring resistance via aneuploidy or extra chromosome copy numbers is something that we published a long time ago, and it just shows that if having extra copies of certain genes helps you grow in the presence of drug, then mutations or changes in the chromosomes that let you have an extra copy of a chromosome or a part of a chromosome that carries those specific genes will allow you to grow better in the drug than your neighbors. And that's actually the second part of evolution of drug resistance. You need a mutation, be it a point mutation or a change in copy number or whatever. And then you need selective pressure. So if there were no drug around, those mutations would be there, but they would never provide an advantage, and they would remain at 1 in 10 million cells or something like that. And in fact, very often the mutations that give you drug resistance give you at least some slight disadvantage relative to the non-mutated cells around you. But if there's drug there and that mutation gives you an advantage in the drug, then all of a sudden you'll be selecting with a very strong selective pressure for those cells that can grow in the drug relative to those who can't. So then the resistant guys become the majority of the population. And that's something that happens not only in fungi and bacteria, also in cancer cells. When we treat with anti-cancer drugs, we also get lots of aneuploidy and lots of aneuploid cells that have acquired the ability to grow in the drug that you're using to treat them.
Okay, so this problem is really very real at the moment. And, it is obviously, we've heard the stories about how there are superbugs coming out in the media and all of this. So tell us about the story about the persistence and the resistance that you have, that you now recently published. Yes.
Yeah. Okay. So I think it's really important to remember that even when you have resistance, some of the cells will still get treated by the drug, but more of them will not than if you have a strain that's not resistant. So strains that are not resistant are strains that are called susceptible or sensitive. And one of the things that we found is that's been known but hasn't been given, I think, enough attention, is that a lot of susceptible isolates that should be treated by the drug, still go on to cause infections that don't get cleared by the drug treatment. That is, they're still causing problems despite the drug treatment, and that's a very big issue. So if you have a lot of infections that are not responding to the drug, then the question is, why is that? And those infections are susceptible, they're not resistant. And that's where we kind of started out with in this study. So it was thought that most of that was because of the host itself, that is the patient. How good is their immune system? How good is their physiology? And that basically that was just an issue of the patient. But we decided to test the hypothesis that this was also a function of the fungus itself, that there were fungi there were specific isolates that would be cleared less well than other isolates, even though all of them were susceptible. So the first thing so this is sort of where that whole story starts. So tolerance is a condition where some of the cells in a population, not all of them, continue growing slowly and not rapidly like resistant cells would in the presence of an antifungal drug. So it can be detected, for example, on Petri plates if you have fungus growing there and it's very slow growth in the presence of the relevant drug, whereas most of the cells in the population don't show up on the Petri plate when you give that drug. And different isolates have different levels of that ability to grow slowly on the plate. And from, let's say, 20% to 90% of the cells in the population could be growing. So the ones that have 20% we'd call low tolerance, and the ones with 80 or 90% would be high tolerance. And we basically hypothesized that because tolerance cells grow slowly, they may be affecting the response to drugs slowly. That is, it may even appear that for the first few days, the patient is responding to the drug. This is something that's seen. But then after a few days, the fungus starts coming back. And we thought that in this case, you would expect that if the reason is tolerance, then you would expect that high tolerance cells would be more likely to come back to grow in the patient or to grow in the presence of the drug than the low tolerance cells. And so that was something we tested, and we also found that tolerant cells, high tolerance cells, were really sensitive to certain inhibitors that, if they were used in combination with the original drug, were able to completely eliminate the tolerance and kill the cells, whereas the original drug doesn't kill the cells, it just stops them from dividing. So we found that there were all these different drugs that could do that, and we then decided that, and in vitro, that is, on Petri plates, we could show that the high tolerance cells were, all of the tolerance was eliminated by those extra drugs, whereas for the low tolerance cells, they weren't really affected very much by the addition of that drug at all. Now, this brings me back to a concept of combination therapy. We only have 3 types of drugs, of drug classes, that we can use against fungal infections. And so one of the more rapid ways to potentially improve antifungal drug efficacy would be to add another drug to the antifungal, allowing it to be more effective. And so what the results that we found showed were that this would be true for high tolerance strains, but not necessarily for low tolerance strains. And so it also suggested that it's really important that you measure the level of tolerance in your strain. This is not something that's normally measured by standard assays, although it's very simple to measure just by extending a standard assay that's done today.
Okay.
So we wanted to test the idea that combination therapy would be more effective than the drug alone against high but not against low tolerance level strains using an animal model, and we collaborated with Jules Eny and Richard Bennett at Brown University to do those experiments in Galeria moth larvae. As the idea had developed on Petri dishes, we also saw in the animal model that low tolerance strains responded better to the drug alone, but the high tolerance strains were the ones that responded to the combination therapy. There's really no benefit to providing the combination of drugs if you had low tolerance. We then also looked at some clinical samples that we got from collaborators, Arnaldo Colombo from Sao Paulo, Brazil, who had collected these isolates from patients that had or had not been able to respond to the single drug, the normal antifungal. And in all of the cases I'm talking about right here, the antifungal is fluconazole, which is the most widely used drug, partly because it has the fewest side effects and it's off patent, so it's not expensive. And what's important there is that all of the isolates from Brazil had exactly the same level of resistance. That is, they were all susceptible to the drug to the same degree. But when we tested their levels of tolerance, we found that the group of strains from different patients who had responded well to the drug initially all had low tolerance. And the group of isolates from the patients who had not responded well to this single fluconazole therapy all had higher, had a significantly higher level of tolerance than the ones that had responded well. So that provided additional support for the idea that it's important to know what tolerance levels are and perhaps to take it into account when designing your antifungal therapies. So now the big biological questions are, and again, I'm a basic biologist, how do you understand how tolerance works? Because it's not a genetic phenomenon like resistance. It's actually due to some of the cells in a clonal population. That is, all the cells in the population are presumably have the same genetics, the same genes, and yet only some of the cells in the population are able to grow. And if you take the ones that grow and ask again who can grow, you'll again see that only some of them grow. So it's not something that is acquired. It's something that's just the ability of these cells to, some of them, to grow in the presence of the drug while others don't. So That means it involves some physiological mechanism where some and not all of the cells are changed. And we're currently trying to understand more about how that works. We've already found that part of the story is that different strains with different levels of tolerance will have different proportions of cells that have a lot of drug in them or not so much drug in them. And so we think the ability to get the drug out of the cell or not let it in the first place is an important um, part of the conundrum. And the second question that may be easier to address is why different isolates may have different levels of tolerance. And we think that could be genetic because these different isolates from different patients have different constellations of genes. And in this case, we also see differences in the levels of drug that they take up as a general rule. In any case, we're at the beginning of the road of understanding all of the physiological implications of this, but we think that it's a really important thing to try and figure out because it has all of these issues of implications for how to treat fungal infections, whether to use combination therapies or and whether to use particular drugs or to choose other ones if you know that a strain is not going to respond as well, let's say, to the chosen drug.
So, in the combination therapies that you're talking about, you have fluconazole. That is obviously something that has already been used quite a lot. And you talked about the other combination would be inhibitors. Have these inhibitors been used in humans before at all? So, I mean, at what stage are we
Yeah.
that we can actually think about, having a clinical trial, for example?
Right. So some of the inhibitors that we used in the Petri plates are more toxic than others, and you wouldn't want to put them in patients. But some of them are fairly commonly used, generally recognized as safe drugs that people take all the time. And so then the thing that needs to be tested is how the combination would affect patients, whether there's any sort of interactions that we'd need to be concerned about between the antifungal drug and the inhibitor. Um, but some of the drugs that we were testing are drugs that are used. I mean, some of them that are more toxic are drugs that are used in anti-cancer therapy, but we also used drugs that are used for depression and anxiety, that sort of thing, that are fairly commonly prescribed in the population. So that actually a very interesting retrospective study would be to look at people who are on those medications anyway and see how they responded relative to others to antifungal therapies if it was needed.
Okay. Thank you so much. It sounds like such an interesting story, and it just feels like you are on the brink of, actually being able to treat these infections that kill so many people all over the world. So can we change gears just a little bit before we
Sure.
end? You have been a scientist in this field for many, many years, and so I just wondered what advice might you have for women who are at the brink of thinking about making a career in STEM?
I think you should do what you love. That's the first thing. So if you really love doing science and doing it, and I mean, STEM is a very broad term. So I'm much more qualified to talk about academic science, and I really love it. I love what I do, and I think I have one of the best jobs in the world because I get to pursue questions that interest me, and to do that at the pace that I set, for the most part. That being said, of course, granting agencies and so forth have ultimate um, oversight on those things, right? So you have to get money to do what you want to do, and there's lots of uh, art as well as science involved in making sure that happens. Um, I think that for women today, it's a whole lot easier than it was 30 years ago when I started out. I mean, when I started out, there was no maternity leave for postdocs at all. And there was no, the maternity leaves even for faculty, because I had my second son before I got tenure. I believe it was 6 weeks. And there was no credit for, having a child and being able to delay tenure decisions, that sort of thing. That came a few years after I did that. I think today there's a lot more awareness of that, and I think that's great. I think that one of the things that we need to be aware of is that um, that women can do pretty much anything in science that they want to do, but you need to have the ability to advocate for yourself. And some of that ability is not always what we've been acculturated to. So, as women, at least when I was growing up, it was not considered proper for women to really be assertive and stand out and call attention to themselves. And I still carry that with me, and I know that I don't, sometimes don't do that as much as maybe would be warranted under a given situation. But we need to realize that on the one hand, that's holding us back. So it's good to develop those skills to advocate for oneself. And on the other hand, there are still the people who will see, men can be fairly aggressive and they usually don't get slammed for it. Whereas women, if they become more aggressive than is expected of them, sometimes that becomes a real negative. And it's kind of a catch-22 that's hard to maneuver. And I think all you can do is be yourself and just, try to get by as best you can under the parameters that you're operating under.
Thank you so much. Judy, it has been such a pleasure to talk to you about this fantastic research that you're doing. Thank you so much for your time.
Thank you, Sheba.